Good afternoon from Israel. Good morning for those who are in that other side of the ocean, and good evening or good night for those who are on far away as Australia. My name is Jorge Diener, and as the director of Hadassah International, it is my pleasure to welcome everybody to this press conference where we are going to showcase and present the incredible and outstanding work of Hadassah Medical Organization's researchers, the incredible work that is represented by the results of Hadassah research applications in front of the Israel Science Foundation and its decisions on the number of awards and the percentage of awards among all Israeli researchers that have been given to 19 researchers at Hadassah, and we will hear from 4 of them today. It is my pleasure to turn over to our incredible and miracle maker, Director of Research and Development at Hadassah Medical Organization, my friend, Professor Eyal Mishani, who, as myself, been in very different places, shares the pride for belonging to this institution that is a leading light in medical research for Israel and for the world. Professor Mishani, please.
Thank you, Jorge. So yes, actually last week we got the information from the Israeli Science Foundation that the Hadassah researchers got 19 grants in different tracks, which is more than 50% of the awards which were awarded by the Israeli Science Foundation to scientists in Israeli hospitals in the field of biomedical medicine. And I must tell you that we were not surprised. We were not surprised since it's not the first time that Hadassah researchers got more than 50% of the awards. And actually we know that when you are combining teaching, research, and clinical activity, you reach excellency. And I think that this is thanks to the spirit of the Hadassah researchers, of innovation, of excellency, and of course of curiosity throughout the years. And actually, I think that the intense and the fruitful collaboration with the Hebrew University also contribute to the success of Hadassah researchers, and of course the investment that the institution is putting in the field of medical sciences. For example, we opened the World Foundation Research Institute for Translational Medicine a few months ago, and we opened a new program to support excellent physicians and to push them to do research by the program Horizon. And actually, we are going to award 2 grants of more than 850,000 shekel in 3 years for the winners. And this program is specifically supporting the physician by guiding them and connecting them with a mentor. We give them absorption grants. We are trying to help them with establishing their own labs. We are going to pay Hadassah, the hospital, to give them free time for research. And of course, we are going to award them with the bridging funds until they will get a competitive fund from the Israeli Science Foundation. So another important activities in the field of clinical trials. You know, this morning I just was informed that comparing to 2019, there were more than 470 applications to the ethics committee for new clinical trials in Hadassah compared to 329 in 2019. And you can imagine that the days of coronavirus worldwide and specifically in Hadassah, in Israel, there are several clinical trials which were open in Hadassah to try to cure the disease and to try to improve the testing. And these new applications are also coming from the industry. We increased it from 130 last year to 167 this year. And last but not least, I must tell you that our success is thanks to the support of HWZOA throughout the years, which support us in doing and encourage us to do research. And of course, Hadassah International in the last 5 years, which are trying to motivate us and also trying to allocate the funds to support our activity and to create a better world. So, as Jorge mentioned today, we choose the 4 leading scientists of Hadassah in different fields. The first one is Professor Higazi. He's the head of division of laboratory and the Department of Clinical Biochemistry at Hadassah University Medical Center. And then follow him will be Professor Goldberg. Is the vice chairman for research in the Department of Radiology, Hadassah Hebrew University. And he's an interventional oncologist, radiologist, and full professor at the Hebrew University. And he's also the chief of the image-guided therapy unit at Hadassah Hebrew University. And I must say that Professor Goldberg made Aliyah 12 years ago from Harvard University, and he joined Hadassah, and we are very lucky to have him with us. Dr. Tal Falick, and this is, I must say that this is kind of miracle for me. Tal joined Hadassah and she started her internship a few months ago. And I met her when she, I think on her 2nd or 3rd day, and she said, I'm going to apply for the Israeli Science Foundation, and I'm going to try to get the grant for a physician scientist. And I say, okay, you have my blessing. And surprisingly, few months later, she was one of the 6 physicians who got the award. And I must tell you, this is amazing. She has a great potential. We believe in her, and we have a commitment to help her throughout her track in Hadassah. And I'm sure that she will reach levels that will show us that the physicians of Hadassah make the change. And last but not least, Dr. Shai Rosenberg, who is a clinician researcher. He studied medicine with mathematics and completed MD and PhD in statistical genetics. He's an expert in neurology and neuro-oncology. And Dr. Rosenberg represents a unique combination of clinical expertise with research in computational biology and artificial intelligence. And I'm excited to hear them all today with you. So, Professor Higazi, please.
Thank you, Eyal, and thank you for inviting me and giving me the opportunity to speak in front of this forum. And good afternoon, according to Israeli time. As Professor Mishani said, I'm the head of the Department of Laboratory Medicine in Hadassah Hospitals, and I'm working on blood clot formation and lysis. If you know, you have heard about the COVID-19 or corona disease, is when patients get sick, they get really sick. And as you know, they can go to need very intensive care and to be under ventilator. One of the reasons having this deterioration in their situation is having increased blood clots, especially in their lungs. And when I heard first time that those patients, they have increased tendency of clotting, so I thought, okay, this is my field. Why I am not going to see what is going there? And I'm working, as I said, a long time ago on the process of clot formation. And we discovered several years ago a protein that is present in the neutrophils, which is white blood cells. And white blood cells are connected to inflammation and viruses. So we thought that this is maybe the best setting to see whether what we are doing in theory, it's really related or happening in coronavirus disease. The protein that we discovered, its name is family of proteins called alpha defensins. And what we try to say to examine in our patients that are hospitalized in Hadassah, whether alpha defensin is increased in those patients, and we find that it's really increased. But what we did before, 1 year ago, before the corona disease began, we just implemented our protein in mice. Mice, they don't have this protein. As you can see, those regular mice, they have very small blood clots, while when we insert our gene, the human gene, in those mice, this is the clot, goes from this side to this side, from this side to this side, and this is the difference in the weight. So our protein increases a clot formation in mice, and we thought that this may be what's going on in corona patients. So we examined our patients, and we saw that there is a very high increase in the concentration of alpha defensin in the corona patients, and this increase in alpha defensins, drawing behind it, increase in the coagulation by measuring its markers in the blood. And here you can see the lung of patients that they died from coronavirus. All of those blood clots are full. This is brown color. This is the alpha defensin. Alpha defensin is present at very high concentrations in the lungs of those patients. Furthermore, you can see that the increase of alpha defensin in several patients in blue, it's become before the red, which is the coagulation, which means that our protein increases first and later become the coagulation. And this is very important because if we can't prevent the increase of the blue line, the alpha defensin, we may be able to prevent the increase in blood clots. And if we can do this, we can save the patients all of those complications that they have in their lungs and the necessity to be under ventilator. And in our earlier studies, we showed this in animals that really we can prevent the increase of alpha defensin by treating those patients with certain medicines, non-medicine called colchicine. And we examined this colchicine in patients of coronavirus, and really we saw that there is very nice improvement in their situations. So other groups in the world did the same thing. There are very recent data showing that really treating patients with colchicine may prevent the formation of blood clots and may improve their situations. What we can say, if really our theory is correct and we can improve it on large scale and big number of patients, we can just convert the coronavirus disease to real or simple flu disease, and we hope that we can do this. And thank you.
Professor, sorry, Dr. Tal Falick.
Can you hear me?
Yes.
Yeah, I'm going to share my screen. 1 second. Okay, can you see the screen?
Yes.
Okay, so first, Eyal, thank you for the warm words. I'm very happy to be at Hadassah. It is really my second home. And hello, everybody, and I hope you enjoyed this short talk. And today I speak with you about the systemic epigenetic changes in injury and cancer. This is one of my projects, and I'm very excited to share with you some of our most interesting findings and future goals. So first we start with some introduction. So every cell in our body contains our whole genome. However, every tissue knows how to function uniquely and has its own set of genes. This is due to the fact that though each tissue has the same DNA sequence within it, each has its own epigenetic signature governing the expression profile. And as you can see here, this DNA, and these are the modification, the flags, and I chose especially DNA methylation, which is very, very well-known epigenetic mark. So these modifications are established during early development, but can undergo dynamic changes due to the different life circumstances. These changes are maintained in the cell and serve as the epigenetic memory mechanism. So using the muscle as our model, we injured one muscle and looked at how the tissue responded. We also looked at how other muscles in the body, not the one that was directly injured, responded. And we found that indeed there were changes in the injured tissue. But most interestingly, we found that other muscles were also affected by the injury. This must mean that a factor circulating in the bloodstream causes this effect. And we also saw that this response signature is maintained long-term after. And when we re-injure the muscle, the response to the second injury and beginning of the healing process is much more rapid. And also not only in the same muscle, but also in other muscles in the body. It is this process that we wish to understand in the hopes of achieving better management of wound healing in the clinical settings. So also tumor formation and the injury process share many similarities in that both are insults to the body that activate the immune system. Therefore, we hope to use the knowledge we gained over regeneration processes to study the systemic effect of the tumor on different organs. These studies will hopefully contribute to improving patients' overall survival and quality of life. And I thank you for your attention.
Thank you, Tal. Dr. Rosenberg, please.
Do you see my screen?
Yes.
Hi, good evening and good day. I'm very happy to be part of the Hadassah family and to be here this evening. And thank you for the support of Professor Mishani and all Hadassah that enables me to do a real combination of clinical work and research, which is extremely important for me. I want to share with you today my research proposal, which related to RNA and machine learning for better precision medicine in cancer. Tumors are caused by mutation in genes, and it appears that every tumor and every patient has a unique mutation profile. Accordingly, if you measure exactly the DNA, the mutation profiles of each patient, we will be able to offer these specific patients a treatment that is personally tailored for him. The advantages of this approach is that according to clinical research, it's a 3 times higher response rate, it almost doubles survival, and it is very simple. Every patient has between 1 to 8 mutation, and we just have to prioritize between them and give a drug. The disadvantages is that it is applicable only to 40 to 70% of patients. The other doesn't have a mutation that is druggable, only 30% response rate, and it is difficult to predict response. There might be 2 patients with the same tumor, with the same mutation, one will respond and live, and the other one will not respond and die. Accordingly, this approach is good, but needs to be improved. And I wanted to propose to do it using RNA. First of all, what is RNA? This is the molecule of the DNA. This is the gene. The cell reads the gene and decides how many molecules of RNA of these specific genes to transcribe. This molecule in time and the level of the molecule mandate how much of the protein of this gene will be translated, and the proteins are the molecule of life that do the job and the processes in the cell. So we will have, by measuring the RNA, a comprehensive genomic view of the patient, 20,000 genes at once. Each gene has its own activity measure, and those measures represent the dynamic state of the tumor and the biological processes. Of course, when we have such a large amount of data, we need to do big data and advanced machine learning methods to analyze it. So how do we plan to use machine learning for this? We are going to collect patients which, according to prior research, we know that they have an active biological process, and we will collect big data on them, clinical data, and of course, RNA data. We are going to repeat this to another group of patients that doesn't have this biological process, according to our prior research, and collect for them the same big data, the RNA and the clinical data, and etc. We will put all this huge amount of data, we'll give it to the computer, and we will ask the computer to use several advanced mathematical, computational, machine learning methods in order to create a model that uses this data to predict whether biological process is active or not. When we will repeat this to many, many important cancer biological processes, we will have a view by using this RNA of which biological processes are active and which are not in each specific tumor, and hopefully we'll be able to provide the patient with better precision medicine. So what are our research goals? First, we want to identify the biological processes in tumors. We want to quantify the activity of those biological processes, and we want to apply them to the tumor that I treat personally in the clinic, the brain tumor. And I'm winning the most severe and deadly brain tumor, glioblastoma, which has life expectancy of a little more than a year and is not curable. We will do it with collaboration with one of the most important research centers of neuro-oncology in the world in Paris, in the Salpêtrière Hospital. And we hope that our study will enable us to apply our findings to the clinic, to offer candidate drugs to patients according to these algorithms, to predict the drug efficacy. And also, if we identify, and we will be able to identify several processes and to rank them, to prioritize them and to select combination therapy of 2 or 3 drugs to provide personally to each patient. And we hope that this will provide the patient with a better precision medicine compared to current advanced practice. Thank you very much.
Thank you, Dr. Rosenberg. Professor Goldberg, please.
Thank you. Just 1 second as I share my screen. Okay. Is my screen being shared?
Yes.
Okay, thank you very much for this opportunity to share some of the cutting-edge space-aged medicine that's happening in the radiology department at Hadassah. And thank you for allowing me to share and explain what we're trying to do in this particular grant. And what we're trying to do is to get rid of potentially pro-cancer effects that we can sometimes see within the types of treatments we do, such as image-guided tumor ablation via micro-RNA inhibition, which I will hopefully be able to explain in just a moment. Before we actually get into what the micro-RNAs do, allow me for a few minutes to explain some of the space-aged medicine that we're performing at Hadassah and throughout the world, in part due to collaborations that include our laboratory. And we're now part of interventional oncology, which is starting to be considered a 4th pillar of cancer care, in addition to medical oncology, radiation therapy, and surgery, particularly because we have techniques that can potentially supplant the need for surgery. If we can place skinny little needles into tumors and apply energy and heat them up, we can potentially destroy them in situ, in place, without surgery. And please allow me to share one example of that in movie form. So here on a CAT scan, I'm a radiologist, we see a small liver tumor, which we once again can appreciate here on this ultrasound as this region over here. It's a viable tumor, as you can see by this bright speckled pattern. In a moment, we're going to see that I placed an electrode into the tumor, apply energy, and you see this white area, meaning that we are destroying by heating the entire tumor. Now we can't see anything because of the fact that it's totally destroyed. And if we look at the follow-up CAT scan, we can see a large area that covers the tumor and essentially enabled us to successfully treat this tumor without requiring surgery. And fortunately, these methods that are in part being pioneered at Hadassah are becoming more and more accepted, to the point that they're now being performed over 100,000 times a year throughout the world, particularly for liver tumors, small kidney tumors, lung, and bone. And this is very exciting translational work from the bench to the bedside, and it works in about 90 to 95% of patients. However, there are times that we see things that we don't want to see, such as explosive tumor growth after the tumor destruction. So here's an example of a case where we had a simple small tumor and we thought we successfully treated it, but 1 month later, one can see all these other arrows, seeing multiple other additional tumors, suggesting that there actually is an increase in a small minority of patients of tumor. We're trying to get rid of tumor, but unfortunately, sometimes there's more tumor there. So we developed animal models to study this at Hadassah, and indeed we showed that under some conditions, if one uses energy such as radio frequency, you get many more tumors in this mouse's liver compared to the control. Fortunately, we're doing additional work combining the therapies with drugs that target various products such as cytokines and proteins that can reduce the tumor growth as well. And it turns out that in a previous ISF grant that was given to us at Hadassah, we looked at some of the various proteins that were associated with this process and noticed various growth factors such as hepatic growth factor and VEGF, a vascular growth factor, that were elevated, and we tried to target them with some level of success. However, it's becoming more apparent that the system is very complicated, and we're not only dealing with proteins, but we're also dealing with other products, such as the ones that Dr. Rosenberg mentioned, RNAs. And in particular, Hadassah is one of the centers, Eitan Galun being one of the leaders in that, as well as many others, that are looking at these small pieces of RNA called micro-RNAs and helping to establish the fact that they can be pro-inflammatory and pro-tumorigenic, meaning that they can contribute to cancer, and they do interact with the various proteins as well. So accordingly, we wanted to look and see if it was only proteins or if these micro-RNAs were also involved. And it turns out that in patients, we did see elevation of several potentially lead candidate micro-RNAs, particularly after tumor ablation. We see elevations of this, but more importantly was the fact that in the patients that had these elevations, those were the patients that went on to produce this tumorigenesis and have bad results, whereas patients who did not have these micro-RNAs tended to have good results. Very similar to the markers, the biomarkers that were mentioned in the first talk, but here for cancer. So what are we going to try to do? We're going to try to understand the roles of these micro-RNAs to get better precision medicine. So in our first aim, we're going to try to actually identify and characterize and understand which micro-RNAs are present after our treatments and see which ones potentially can contribute to the bad effects that we want to eliminate of tumorigenesis. In aim 2, we actually are going to try to figure out how these various microRNAs interact with the proteins and the cells that are present. And most importantly, in aim 3, we're going to try, once we understand these pathways, to target these key microRNAs and with various drugs to try to eliminate the negatives and get even better results with our therapies. And thank you very much. A picture from my laboratory overlooking Ankara.
Thank you, Professor Goldberg. Jorge? We can't hear you.
Thank you, everybody. I want to thank Professor Mishani and all of you, and each one of you, on behalf of Hadassah International and all the friends, supporters, followers of Hadassah. You know, it's one thing I want to make sure those who are listening and watching this press conference know is that it's remarkable to think that in a year like this, there are people at Hadassah Medical Organization here in Jerusalem that while the hospital is still been coping with an influx and flow of both COVID-19 patients, taking a big part of the burden right now of having COVID-19 patients coming through our emergency, getting into the wards, and at the same time, dealing with all those patients, regular patients who have returned to the hospital, understanding that we have built a structure in the hospital that enables those who have all kind of other diseases, the regular patients, those who come to your clinics, to come because they know that it's safe to be at Hadassah and that you will not get infected by COVID, that you can still get treatment. And in the middle of all that storm and crisis, we have our researchers who also see patients. We have our researchers, you and your colleagues, doing this incredible work that is, when I look at this, and I think when everybody looks at this, what we might not understand all the technicalities of the research you are doing, but if something comes clear is that Hadassah Medical Organization is a magnet for people like you who came to do your research at Hadassah, to be professionals, medical professionals at Hadassah, but it's also an incubator of the knowledge that will bring cures to disease now and in the future. And if it's coming through a combination of machine learning, like you presented, and understanding better genetics, and looking at the process of how we stop blood clots from creating the catastrophe that creates a virus like a COVID-19, or if it looks at the radiological elements that can make that difference in curing the ongoing and chronic and terrible diseases that humanity faces. I think that summarizes the impact that has the work that you are all doing. And it explains why the Israeli entity that has to decide among hundreds or thousands of Israeli researchers who want to receive these grants, why they have chosen 75% of the researchers and physician researchers, 75% of the grants to be awarded to Hadassah researchers, and around 50% of all the grants given in Israel given to Hadassah researchers. We are proud. We are happy we can share this remarkable milestone for Hadassah with the world, but we are more proud that from Jerusalem, from Hadassah, we can share this knowledge to the world and bring cure and better treatments for humanity. Thank you so much for sharing your knowledge, for the work you do. And thank you all those participants that have been at this event for being part of our Hadassah network and for the Hadassah family. Thank you very much.